You're gonna live tweet it?
The followers make it worth it.
Oh my god, I'll retweet you. I was probably gonna tweet at least one.
Let me see if it works. Oh, perfect. Mike, you're on station. It's now.
Amazing.
We're good.
Cool?
Yeah, just leave it.
Yeah. It doesn't need the volume up or anything. Should I
No,
mute it?
there's no.
Should I mute it so that my sounds don't come through?
So that,
Like, if I get emails or anything, they don't go off.
I guess that should be fine. Let me make sure actually I can hear it through my computer.
Okay.
Let me make sure I can.
Great. Okay. Perfect.
Okay,
Cool.
Thank
I'm
you.
gonna sit, no props, appropriately far away.
What's up, dude?
hi Eric.
Oh, hey.
How are you?
Sorry.
Yeah.
It's fine. I feel like professional sites are here.
No, this is before. So now it's 6. Oh, yeah, my God. I told them numerous times, like, tell her.
More than Colorado. I always hear something out there. Okay. It's never. Yeah. I mean, I'm sorry. It's like we had a practice. We did like a pilot learning series. And what I
Oh, that's right.
It was like the whole as we were writing.
That's unexpected.
It results in the same type of.
Oh, I think so.
I know, but I think it specifically was my. Like on, not a person, but on valve. It creates very similar.
No, yeah, the best thing we're actually doing today.
No, I think it's really.
I'll often see a focus of that is distinct.
Because colors, I mean, that brown stuff is so generic, whereas like resin is very distinct.
It's like the patterns are very not like travel along the desert, like the.
I think Brooklyn's up in the test, she's very.
Immediately over the dogs.
I mean, that's what I see. That's really possible to be like, years ago. Before, it was like, 2014, my first year of grad. No, 2015, it was fine. It was for the iteration of the grant that was successful. I never not believe Oh, all of them sound great. I don't think so. That would be something I should listen.
We're hoping to inoculate cost options in the next.
You know.
Actually, I love her.
Yeah.
Honestly, I'm happy to
Yeah.
I like that. Yeah. Over the course of the season, like the Browns sponsor
Oh, my goodness.
Well, they are different.
Hey, pretty well.
Alright.
I did, and I have something very special for you. I hope so. Let me guess, that's Mike.
That is fine.
Mike, can I come meet you, Mike?
Oh, yes.
Hi, I'm Corbin Jones. I've heard a lot about you.
Nice to meet you.
All of it good.
Thanks.
I suspect that's. They kind of have that vibe. All right, I got some questions on senior
I can't hear you.
You want, I swear. I don't know why. Yeah. You know, my wife follows her. Yes. On Twitter. She didn't know who you were. She did not know who you were. I believe it was because she sees it. Yeah, some connection. Yeah. You probably have fun. Yeah, that was, I think, how she did. We like put some on like science Twitter. Yeah, I mentioned something the other day. She said, "Oh yeah, I saw it on Twitter." Yeah. When we were coordinating last week, when we were discussing how we were coordinating today. I was giant conference. Yeah. It's just reminding me, Kayleigh. So we had our annual, and I had
Most important thing.
Kayleigh texted me, "Are you coming to lunch?"
Ah,
Oh, that's what that means. I was working at Hopkins.
you were at home?
Yeah, I was too. I dashed over. But I'm here. I'm good.
Oh. Let's put my water like
So James, I feel that because I almost wore a very bright jeans. I said, "Oh,
I had my coat
That's good. She's on my committee.
yeah."
Charles, can I give this to you? Hold on to. I'm gonna forget where I put it.
Oh, that reminds me. So we staying in here after this or the
I reserved this room, if that's
Great.
okay.
It is. I just couldn't remember.
Yeah.
I'll just remind the committee members afterwards
Yeah.
to stay here.
Yeah. A while we were told
All right, just one more.
Yeah. Susie's in her lab. She said she's on the way.
So,
Susie's in Beth's lab, so she said Beth's on the way.
You're there.
we still have one committee member coming, so.
Yeah.
You can talk for a minute.
Tell jokes, Charles.
One more person coming.
Tell jokes. Warm the audience up. No?
I'll leave it to you.
What's a fisherman's favorite game show? Name that tuna.
Yes.
You're gonna look great.
There's Beth.
It'll look great. No. Find out.
Yeah.
Wow, congratulations. Well, yeah, too much.
Okay, welcome. Thank you for coming. So, before starting at UNC, Kayleigh graduated cum laude from Amherst College, and then spent 2 years working at Tufts University and as a research lab technician. When she started here at UNC, I had high expectations. In fact, in retrospect, my expectations were totally unreasonable for anyone to have for any student. I didn't, yeah, anyway. But at the time, I was really excited to shift my group's research to include research on the microbiome. And to do that, I needed someone who could lead our group in applying genetic sequencing. And my expectations for that person, Kayleigh, were a bit naive because I am a field ecologist who has never run a PCR or even a gel. Yes, so anyway, with my own hands. So anyway, my expectations were a bit ridiculous, yet Kayleigh more than exceeded them. And more importantly than my silly expectations, she developed and executed her own independent scientific vision, which is what doing a PhD is about. And so she began by critically examining one of the methods that she plans to employ at RNA sequencing. And as a first author with Corbin Jones as her sole co-author, i.e. without me, which is great.
Thanks, Charles.
She published a paper developing a computational method to better apply RNA sequencing to non-model organisms. After that, or I guess simultaneously, she published a synthesis paper, a conceptual synthesis paper, integrating approaches to microbial interactions across 2 levels of biological organization, the host individual and host population. She won't be presenting either of those 2 published works today. She'll actually be focusing on 3 chapters that she's preparing for submission to journals. She completed all this in just less than 5 years, a timeline which was facilitated in part, well, partly by her extremely hard work, but also by her winning an NSF predoctoral fellowship, graduate research fellowship. While for most students, one of the main benefits of a fellowship is not having to teach, Kayleigh actually loved to teach. This presented a bit of a quandary, and in fact, she sought out opportunities to teach even while on her fellowship. And most notably, last fall, she volunteered through an internship to serve as instructor of record for a section of general biology over at Meredith College in Raleigh, which is quite a substantial time commitments, in addition to doing all of her research and continuing to make great progress on that. So yeah, here at UNC, Kayleigh's been a great mentor for undergraduates, more broadly, and exemplary member of the research group, and really an outstanding contributor to the UNC community, for which she was inducted into the Frank Porter Graham Graduate Student Honor Society. All right, so in August, she'll be starting a postdoc with Dustin Brisson at University of Pennsylvania in Philadelphia, and they'll be very lucky to have her. And so a final quality of Kayleigh's is that she's very modest and humble. And so now that I've probably thoroughly embarrassed her by shining a spotlight on her accomplishments, I'll let her tell you about her research.
Thank you for that generous introduction, and thank you for coming today. Organisms live within ecological communities, which are assemblages of populations of different species that interact directly and indirectly within a defined geographic area. Interactions amongst the species that comprise communities form the basis for many ecosystems' properties and processes, and the nature of these interactions can vary depending on environmental conditions in which they occur. And while difficult to quantify and measure, these interactions among free-living species have been studied extensively within ecology. However, these free-living organisms, ranging from plants to mammals, are host to a diverse set of mutualistic, commensal, and parasitic microbes, referred to collectively as the microbiome. The microbiome is also considered a community, as the diverse microbes of the microbiome, whether they be bacteria, fungi, or viruses, interact with each other as they live in, on, and around their hosts. Within the microbiome, parasites are among the best studied microbes due to their negative impacts on their hosts. For example, the fungal parasite Rhizoctonia solani causes the symptoms shown in this circle and can decimate populations of the grass host tall fescue. Infectious disease has historically been studied in a one host, one parasite framework, but the field is now appreciating the role and complexity of within host microbial interactions. So in this system, while Rhizoctonia is a major parasite of tall fescue, surveys done of local populations of this host, just down in Duke Forest by the lab, have shown that often individuals of fescue are infected by more than one fungal species at a time, whether they be other parasites or a potential mutualist of the host. And each of these fungi can potentially interact with Rhizoctonia. And as we've come to appreciate the microbiome of plants and other organisms, an important frontier is investigating how important are interactions among microbes within shared hosts, and specifically, how do they affect parasites and the diseases that they cause. So what might happen when microbes infect the same host at the same time? Well, they may not interact at all, perhaps if they infect different host organs or require different host resources. But if we assume that they do interact, they may do so directly, perhaps through a physical interaction or through chemical secretion. They may do so more indirectly through the host, maybe by competing for host resources or through the host's immune responses. And these different types of interactions, both direct and host mediated, can lead to different outcomes, ranging from facilitation to inhibition. A key question in infection biology is how do these interactions shape parasites in both the near and long term? In studies of near-term responses of parasites to microbial interactions can include comparing parasite growth rates and subsequent disease severity caused by these parasites as they grow within a host related to these microbial interactions. And evidence for such near-term responses to these interactions have been shown throughout a number of systems. In the long term, studies may look at parasite transmission rates, spatial and temporal patterns of parasite abundances, and the ability of species to co-occur as that relates to microbial interactions. And again, while perhaps less represented in the field, there is some evidence of these long-term responses of within-host microbial interactions across a number of systems. I'm motivated to understand these near-term and long-term effects of within-host microbial interactions on disease, and to do this, I focus on parasites at two levels. The individual level, such as a parasite here shown in white, growing within an individual leaf, interacting with co-infecting microbes on that leaf, here shown by different colored triangles, up to the population level, such as different individuals growing across a number of host individuals interacting with co-infecting microbes. And I've been exploring the ecological and evolutionary, something popping up on my computer. I've been exploring the ecological and evolutionary implications of within-host microbial interactions by focusing on a plant parasite system at both of these levels, as well as exploring the community context of this system. And today I'll be starting out by talking about one of my population level studies. Returning to what we know from free living species, we know that mutualistic interactions are ubiquitous in nature. This type of species interaction involves the exchange of goods or services between two species. And while ubiquitous, mutualisms involve many different kinds of organisms, highly variable types of services exchanged, and varying mechanisms by which these exchanges occur. A classic example shown here is the interaction between a plant and its pollinator. One type of mutualism is a defensive mutualism, in which one species receives protection against predators or parasites. Examples of such include the one shown here of cleaner fish eating off the surface of a grouper, and in the parasite removing ectoparasites of that grouper. And while mutualistic interactions have been studied of free living species for decades within ecology, as more attention has been placed on the importance of the microbiome, such interactions have been studied between host organisms and their microbes. Defensive microbial mutualists can dramatically impact the survivorship, growth, and reproduction of parasites infecting the same host individual. An example shown here is work led by Betsy Arnold at the University of Arizona that showed that the fungal community of the cacao plant, the plant that gives us chocolate, can protect the host against parasites that cause diseases like the one shown in this picture, black pod rot. And while there are many studies investigating the impacts of mutualists on parasites within host individuals, the connection to the impacts of mutualists on parasite transmission across host individuals remains unclear. And this is important to consider because populations are likely more complicated than just the sum of a number of individuals, and what happens in individuals may not clearly scale up to a population. So with that in mind, I explored this connection with a field mesocosm experiment. So all the fieldwork that I'll be presenting on today occurred in Duke Forest. The fieldwork I'll be about to present occurred in this old field right here that's just 20 minutes or so up the road. And most of the green that you're seeing in this picture is tall fescue, the host that I'll be focusing on in all the empirical work of my dissertation. And this proved to be a really excellent system to work in because surveys that we've done of this system have shown that tall fescue is often infected by more than one fungal species at a time. Rhizoctonia solani is a fungal parasite. It causes the symptoms shown here, and when it parasitizes a plant, it parasitizes as a necrotroph, meaning that in order to extract resources, it needs to kill host cells. So in this picture, Rhizoctonia is actively spreading in this dark part of the lesion, and the white part is dead tissue it's leaving behind as it grows through the plant. In contrast, Epicloe xenophiala is a systemic endophyte, meaning that it grows asymptomatically in its host, and in this case, it's often considered defensive, as it's been shown to protect its host against herbivory and different infectious diseases. And in this case, unlike Rhizoctonia, which like a lot of parasites is transmitted from individual to individual within a generation, Epicloe xenophiala is vertically transmitted, meaning that it's transmitted between generations through the seed. And so these two different fungal species co-occur in this tall fescue population. Surveys of this fescue population that we've done showed that 75 to 99% of individuals in the field are infected with epicloe. And this population experiences a pretty typical epidemic of the parasite Rhizoctonia. So on the y-axis here, we have the proportion of leaves that we surveyed that were infected with Rhizoctonia, and you can see that it peaks in the late summer, early fall. And knowing that these two fungi co-occur in this system, we were interested in asking, how does the defensive mutualist affect spread of Rhizoctonia, this parasite? I did this in collaboration with Brendan Wheeler, who recently graduated. And so we set up this field mesocosm experiment in that field population. And as you can see, it was really high tech. It involved a lot of kiddie pools. Sold out 2 different hardware stores of all the kiddie pools that they have. And filled each of them with soil and then planted plants according to this matrix. And within each of these populations that we created, plants were either all inoculated with epicloe, this potential defensive mutualist, or all lacking the defensive mutualist. And then we inoculated the central plant with the parasite, Rhizoctonia, and after inoculating the parasite, we tracked spread of it from leaf to leaf within that central plant, as well as to the surrounding plants, which were planted at 3 different distances away from that central plant. And before I jump into my data, I kind of want to digest what I'll be showing you. So on the x-axis, we'll have the days after we inoculated the population with the parasite, and on the y-axis, parasite prevalence. And what I mean by parasite prevalence is the proportion of leaves in which Rhizoctonia was present. So here we're considering leaves as host individuals instead of plants because the fungal infections are pretty localized to the leaf. So on this plot here of my actual data, the different points are the observations of the different populations, and the lines show the trends across these epicloe treatments. And while it looks like these different treatments are differentiated in some way, there was actually no significant effect of epicloe treatment on parasite prevalence over time. However, when you considered each individual survey date, there was one point at which these 2 treatments were statistically different in the parasite prevalence, which corresponded to the modeled peak parasite prevalence according to these curves. And in fact, when we took the individual peaks of each of the prevalences of each of these populations in my experiment, epicloe did have a significant effect, where populations in which epicloe was present had a higher peak parasite prevalence than the populations in which epicloe was absent. Now I want to take a second here to explain that this was not expected. So epicloe we thought was a defensive mutualist, and so we were surprised to see that when it was present, we actually were getting more individuals infected with Rhizoctonia at the peak of the epidemic. It's a little bit confusing. And so we wanted to interrogate what might be going on in this system. Now I should say that there was imperfect spread of epicloe to above ground tissue. So 100% of the seeds that we used to plant these epicloe inoculated populations were in fact inoculated with epicloe. However, at the end of the experiment, when we harvested all the plants and brought them back, we performed an immunoblot assay which tests for the presence of epicloe, where the dark pink indicates that it's present and the light color indicates that it's absent. And what we found is that only 42% of the plants that we inoculated with epicloe actually had it in above ground tissue, which resulted in these populations ranging from 15 to 69% infected with epicloe. So as we were considering what else might be going on in this system, we focused on different levels, including what's going on at the individual plant level. We actually had to consider 3 different categories, not 2. We had the plants that epicloe was absent because we never put it there. We had the plants in which epicloe was infected, both at the seed and at the in above ground tissue. And these plants in which epicloe was absent, but it had been inoculated with epicloe. So looking at those 3 categories, again, we harvested these plants at the end of the experiment, and we measured above ground biomass. And we found that epicloe infected, so these truly infected individuals, had higher biomass than the 2 other groups of plants, which you can see with the blue bar being higher than the orange and the green bars. And so This translated to epicloe inoculated populations trending towards having this higher population level biomass than epicloe-free populations. But I should say that statistical support for this was pretty weak. And so given this, epicloe is a mutualist. It's resulting in more biomass production for its host. And so we wondered, could this mutualist have a secondary effect for the host population? Namely, does higher biomass correlate with higher host density? So in this case, I had mentioned that host individuals are individual leaves. And the reason why we were wondering if higher biomass might correlate with host density is because under density-dependent transmission, the per capita contact rate between susceptible and infected individuals depends on population density. And so a simplified relationship here is such that under density-dependent transmission, as population density increases, transmission rate would be expected to also increase. So we were wondering if we had this higher biomass and it correlated with higher host density, maybe through this increase in contact rate between susceptible and infected individuals, we're getting that higher P. parasite prevalence. And so I should say that in this iteration of the experiment, which happened last year, we didn't count the total number of leaves per plant across the whole experiment. But we did, and I should say Brandon did, in 2017, which was a lot of leaves, but he did do it. And so we used data from 2017 to see if this correlation held for a previous iteration of this experiment. And at the plant level, on the x-axis, we have above ground biomass, and on the y-axis, we have the number of leaves within a plant. We found that there was a positive correlation. As biomass increased, the number of leaves within a plant also increased. And at the population level, so considering the population level above ground biomass and the population level number of leaves, we saw a similar trend, though again, it has weak statistical support at that level. But given this relationship, we wondered if we included biomass in our models for P. parasite prevalence, does it account for this effect epicloe is having on P. parasite prevalence? And when we did that, so on the x-axis here, we have population above ground biomass, so putting together all the biomass of all the individuals in a given population. And on the y-axis, peak rhizoctonia prevalence. Each point is an individual population. And what you can see is that as above ground biomass increases, the peak rhizoctonia prevalence does increase, which may be consistent with density-dependent transmission. However, epicloe did have an effect beyond this effect on biomass on P. parasite prevalence, which we can see with the two lines, blue and orange, still being separate from each other. So while biomass might be part of the picture, it didn't count fully for this effect epicloe is having on parasite prevalence. So I want to take a second to say that population level studies of within host microbial interactions are generally underrepresented. Most studies are within host individuals under lab conditions, which makes a lot of sense. It's a lot easier to do that. You have more control over what microbes are present. You're introducing them into the system. But it's critical to understand how impacts of interactions scale. In this case, we thought it was a defensive mutualist, and we weren't getting the expected results. And so field mesocosm experiments like this facilitate such studies under field conditions. I should say that this is the first, I think I proposed this to Charles my first year of grad school, and it was the first type of experiment in which we did kind of look at this, use this type of experiment in this system. And it allowed us to learn what was going on at different levels. Specifically, at the plant level, epicloe increased biomass production. But at the population level, epicloe was associated with this increased P. parasite prevalence. And while biomass might be contributing to that in some way, it wasn't accounting for all of this effect. And so, kind of sitting back and thinking about these results, we wondered, and maybe you're wondering it too, is epicloe actually a defensive mutualist? It's kind of surprising. So we found that it's a mutualist, so meaning it is gaining a benefit. But if that benefit isn't that it's protecting it against a parasite or a predator, it's not a defensive mutualist. So we were wondering if epicloe is truly a defensive mutualist with regard to rhizoctonia specifically. And more generally, we were wondering if individual parasites respond to within-host microbial interactions. So we did leverage the advantages of lab conditions. So lab conditions allow us to have more control, and we interrogated this interaction between Epicloe and Rhizoctonia within host individuals. And so we did this with a factorial design where in a growth chamber under controlled conditions, we had different plants that were within this factorial design of Epicloe inoculation, so infected or not infected, and Rhizoctonia inoculation. So it was inoculated with one of 3 strains of Rhizoctonia or mock inoculated just to see that we weren't getting any cross-contamination between hosts. And I should say that in the previous experiment, I had this third category of plants that were inoculated, but it wasn't present in above ground tissue. That category was omitted from analyses here since we're just looking at the host individual, not the population. So my data will look like this. So on the x-axis, we'll have days after we inoculated with Rhizoctonia, and on the y-axis, the regular measurements that we took of these plants. So disease severity, here measured by lesion size. So the previous experiment was just looking at the presence absence of Rhizoctonia on a leaf. Here we're actually looking at lesion size. And if we expected Epicloe to act as a defensive mutualist, we might expect for the growth rate of Rhizoctonia, this parasite to be lower when Epicloe is present. In fact, what we saw was that was very much not the case. So the faint lines are the measurements we took of individual leaves in this experiment, and the trends are in the bolded lines here. And you can see those bolded lines are really right on top of each other. There was no significant effect of Epicloe on growth rate of Rhizoctonia within a host. So this suggests that in fact Epicloe is likely not a defensive mutualist as it relates to Rhizoctonia. And I mentioned before that Epicloe has been studied in its symbiosis with tall fescue, and it has been shown to be a defensive mutualist against other parasites, which is suggestive that this interaction might be specific to different parasite species. So we interrogated this pairwise interaction between Rhizoctonia and Epicloe. But I mentioned that we had all these surveys of this field site, and they certainly weren't the only fungi occurring in this system. So we wanted to further understand other interactions that might be going on. Colototricum cereale is another parasite, and Rhizoctonia and Colototricum co-occur within this population of tall fescue. So here I'm showing you that epidemic curve of Rhizoctonia I showed you earlier, and Colototricum co-occurs in this population, actually at higher levels than Rhizoctonia. So it's around 60% of the leaves that we surveyed actually having symptoms of Colototricum. And not only do Colototricum and Rhizoctonia co-occur in this population, they've also been observed in co-infection on individual tall fescue leaves. So in green, I added the proportion of leaves infected with both parasites. And you can see that that's really driven by that Rhizoctonia epidemic that occurs as it goes up and down in the same fashion. And so to talk about them a little bit more, Rhizoctonia and Colototricum are fungal parasites. And I already mentioned that Rhizoctonia is a necrotroph, meaning that it kills host cells in order to extract resources. I'm going to refer to this a number of times in the rest of this talk, so I figured I should symbolize it. So it's going to be symbolized with this emoji, whereas Colototricum is a hemibiotrope. So it actually infects hosts in 2 different phases. It initially keeps host cells alive and then switches to killing host cells in order to extract resources. So I'm going to symbolize it with these emojis. So given these different feeding strategies, we might hypothesize different interactions between these parasites. When Colototricum initially infects a host, it infects as what's called a biotrope, so keeping host cells alive, whereas Rhizoctonia is a necrotroph. And based on these different feeding strategies and associated immune responses to each of these parasites, we would hypothesize that Colototricum would facilitate Rhizoctonia, and Rhizoctonia would inhibit Colototricum. When the infections progress and Colototricum switches into the same type of feeding strategy as Rhizoctonia, and we would expect them to mutually inhibit each other as they feed on the plant in the same way. And so given these different hypothesized interactions, I worked with another undergraduate who worked with me a few years ago, Anita Simha, to explore how co-infection with Kayleigh affects Rhizoctonia's ability to cause disease. We were specifically interested in how infection sequence might modulate this interaction. So we performed another inoculation study and another growth chamber experiment. And in this case, we weren't looking at Epicloe, we were just looking at the parasites. And we had plants that we inoculated with just Rhizoctonia, with Rhizoctonia and Kayleigh simultaneously, and with both parasites but inoculated sequentially. So one treatment in which Kayleigh came first, and one treatment in which Rhizoctonia came first. And following these inoculations, we tracked the presence-absence of symptoms caused by each parasite, as well as the lesion size caused by each parasite. And to kind of jump right into my results, we saw that co-infection with Kayleigh resulted in an increase in Rhizoctonia growth rate. So you can see that all of the 3 co-infection treatments, regardless of the infection sequence, had a higher growth rate than when Rhizoctonia was inoculated on its own. So I should say that we're looking at Rhizoctonia's growth rate, not Kayleigh. And so to think about those specific results, the biggest increase happened when these parasites were inoculated simultaneously. So the orange line is when Kayleigh and Rhizoctonia were inoculated at the same time, whereas the yellow and green lines are showing you these different sequential treatments, which each had a more intermediate increase in growth rate of Rhizoctonia. And so co-infection, regardless of infection sequence, did always increase Rhizoctonia growth rate. However, the magnitude of that increase depended on infection sequence. But it might not be that order of arrival mattered so much as the difference between simultaneous inoculation and sequential inoculation. And so to think about the results that I've shown you so far, I showed you that Epicloe had no effect, or no effect that I could detect, on the growth of Rhizoctonia within a host, whereas Kayleigh facilitated the growth of Rhizoctonia. And taking some results from a lab mate, Fletcher Halliday and collaborators, from a field experiment, they found that Epicloe inhibited Kayleigh. They did also find that Epicloe had no effect on Rhizoctonia, but they found a different interaction between Kayleigh and Rhizoctonia, which may suggest that this interaction varies with environmental context, given that his experiment was in the field and my experiment was in a lab. This adds to mounting evidence that interactions can differ in direction and magnitude between field versus lab conditions. This isn't necessarily surprising. Those are very different environments, both abiotic and biotic. And while the results of field experiments are incredibly important for us to know the natural dynamics of a disease system, it may be neglecting microbes that are difficult to detect or that don't cause an obvious effect on host phenotype, like a parasite inherently does. And so we wanted to leverage the advantages of lab conditions to interrogate more complex interactions that aren't just pairwise interactions between different components of this community. And so when I think about complex interactions, I'm thinking specifically about higher order interactions. So indirect interactions among species through effects on intermediate species. So returning to this interaction web I showed you before, and taking away the interaction that we didn't have results that supported that there was a detectable effect, we have this interaction chain, if you will. And you can see that Epicloe inhibits Kayleigh facilitates Rhizoctonia, and so you might expect that Epicloe indirectly affects Rhizoctonia through its effect on Kayleigh. And with a final inoculation experiment, I explored if the interaction between Epicloe and Kayleigh affects the growth of Rhizoctonia. And we did this with a factorial design where we had Epicloe inoculation again, and in this case, parasite inoculation included plants that were inoculated with just Rhizoctonia, just Kayleigh, or co-infected with or co-inoculated with both parasites. And I should say in this case, we didn't consider infection sequence, so the co-infection treatment was simultaneous, especially given that that was when the biggest increase in growth rate happened. And so to kind of walk through what we might expect, I already showed you that when Rhizoctonia is inoculated on its own, has a lower growth rate than when it was inoculated with Kayleigh. But this is all in the absence of epicloe. And returning to this interaction chain, given that epicloe inhibits Kayleigh, we might expect that epicloe might limit the ability of Kayleigh to facilitate rhizoctonia. And so instead, what we thought might happen when epicloe was present was that that increase would either be gone or lower. So to walk through my results here, so in blue we have when rhizoctonia was inoculated on its own, and orange is when it was inoculated along with Kayleigh. And you can see that the orange lines are always above the blue lines, so we were very excited to see that that result, that when rhizoctonia was inoculated along with Kayleigh, we found that it had an increase in growth rate. However, when epicloe was present, we didn't see a change, well, we didn't see a significant change in growth rate in the way that we expected. We thought that it might be lower, but in the dashed line, you can see that if anything, it's higher, though this had pretty weak statistical support for that three-way interaction. And so rhizoctonia disease severity was still higher under co-infection with Kayleigh on epicloe-infected leaves. And so we wanted to interrogate this a little bit more, including the not obvious organism, but the other organism in this interaction, the host. So this is all happening within a leaf. And so we wanted to know how the host is responding to these different treatments. And so here's a survival analysis. So on the y-axis, we have the days after inoculation, and we'll think about the y-axis as the proportion surviving or predicted to survive at different points along this experiment. So in the dashed lines is when epicloe was absent. So when epicloe was absent, there was no effect of parasite co-infection on survival of individual leaves. So we're looking at individual leaves, not plants. However, when epicloe was present, which is shown in the solid lines, we saw that leaves that were co-inoculated with both parasites had significantly lower survival across the experiment than leaves that were inoculated with just rhizoctonia. So the host is certainly responding to this, to the presence of all three fungi. And this was complemented with, after the experiment, when we harvested the plants and measured above ground biomass, the only treatment in which all three fungal species were present also had lower biomass compared to all of the other groups. And so thinking about all of this together, we were wondering if this is evidence for higher order interactions in the system. And so returning to what I had expected, because epicloe was thought to inhibit Kayleigh, I thought that in the presence of epicloe, this facilitative effect would be lower. What we found was that in epicloe-infected leaves, rhizoctonia still had a higher growth rate in co-infection with Kayleigh, and if anything, had an even higher growth rate. Additionally, these leaves that were co-inoculated with both parasites in the presence of epicloe had lower survival than the leaves that were inoculated with just rhizoctonia. And then in epicloe-infected plants, there was lower biomass. So while these interactions weren't happening in the way that we predicted, this data does suggest that considering these more complex interactions is critical to thinking about disease dynamics. Disease isn't just a function of parasites, it's parasites, hosts, and the environment, and what we're showing here is that the host is responding to when all three fungi are present. And so while higher order interactions may not be occurring in the way that I predicted, it certainly suggests that including more complex interactions is critical for us to understand what's going on. So to kind of summarize this part of my talk, systemic endophytes don't always suppress progression of parasites. So epicloe didn't have a detectable effect on rhizoctonia, which might suggest that this effect of epicloe as a defensive mutualist is parasite species specific. Parasites can affect the growth of co-infecting parasites. So in this case, Kayleigh increased the growth rate of rhizoctonia, and the magnitude of that increase depended on infection sequence. And finally, higher order interactions, or perhaps more generally complex interactions, may be important in determining disease outcomes. So when all three fungi were present, we saw that Rhizoctonia still had this increasing growth rate, potentially even more of an increasing growth rate, and that leaves that were inoculated with all three had lower survival and lower biomass. So most studies of within-host microbial interactions are pairwise interactions between parasites. This may be neglecting a couple of things. Pairwise interactions are only considering two components at a time, but these interactions certainly aren't happening in a vacuum, and so we may be missing important other interactions that are included here. Additionally, by only including parasites, we might be neglecting microbes if they don't affect host phenotype or are otherwise hard to detect. So the experiments that I just conducted were looking at 3 different fungi in the system, but they certainly aren't the only fungi going on here. So I was interested in this kind of final part of my talk in what the relationship is between the host-associated fungal community, not just these 3 fungi, and disease. So I already talked about how different microbes may interact with each other. And when we consider how the parasite might interact with the whole microbiome, I'll be showing you time along the bottom of this slide, and I'll be symbolizing a microbiome with a black square, these cartoons. And so if a parasite tries to invade the host, the microbiome might affect susceptibility to that parasite, perhaps resisting the parasite, so protecting its host against infection from the parasite. But if it does allow the parasite to invade, that parasite can then go on to have an effect on the microbiome, perhaps affecting the diversity or composition of that community. And so not only do parasites affect the microbiome, but the microbiome affects parasites. So I was interested in what the relationship is between the host-associated fungal community and disease. And so I went out and was looking for these 2 fungal parasites we talked about already, Rhizoctonia and Kayleigh, as well as a 3rd parasite that occurs in this system, Puccinia coronata, or rust. And so I already talked about Rhizoctonia and Kayleigh as in the context of their feeding strategies, where Rhizoctonia kills host cells to extract resources, and Kayleigh had this 2-phase infection process. Puccinia coronata is a biotrope, so throughout its whole disease cycle, it keeps cells alive in order to extract resources. So each of these 3 parasites have different feeding strategies on the host. And so given these differences, and as I was thinking about what I might expect in this experiment, necrotrophs are modifying the host environment. So to return to this symptom, it's creating dead tissue on the leaf, dead necrotic tissue as it grows throughout a leaf. And so given that niche modification explains that species change the type is when species change types of niches available within a site, and consequently the identities of species that can colonize the community, I hypothesized that fungal communities of hosts damaged by parasites that modify the host environment might be distinct from the fungal communities of hosts that are asymptomatic. And so putting that in the context of these feeding strategies, I thought that necrotrophs, which are creating this necrotic tissue, might be associated with distinct communities compared to hosts that are asymptomatic. Whereas I might not expect the same thing of hosts damaged with biotrophs, given that they don't modify the environment in the same way. So we went out and did, an observational study or a sequencing survey where we went out and collected samples along transects of this field that's part of Duke Forest. I should note that this is a different field from the field you saw before. And at each site along each transect, we collected different types of leaf segments. We had segments that were asymptomatic, meaning that we couldn't see any signs of disease that we took a segment from. And then we had leaves that were symptomatic of each of the 3 fungal parasites. Here I'm just showing one. And from each of these symptomatic leaves, we took a segment that had the symptom on it and a segment that did not. And so we brought all of these leaves back into the lab, extracted DNA, and amplified the ITS region. So the ITS region is the internal transcribed spacer region, and it's a part of the fungal genome that's thought to be conserved between species and different, conserved within species and different between species, and allow us to investigate whole microbial diversity and determine the relative quantity of community members and environmental samples. And the part of this region that I amplified allows us to generally get down to the genus level. So before I go into my results, it's critical to first understand how I quantify diversity. Diversity indicators are differentially sensitive to rare and abundant taxa, and the result of such comparisons may depend on the choice of index. We compared the diversity of fungal communities in these different groups with the Hills series of diversity. So this series consists of 3 numbers, N0, which is simply the number of taxa in a sample, N1 and N2, which are transformations of commonly used diversity indices. And these transformations allow all 3 numbers to have the same unit of effective number of taxa. Further, N1 and N2 put progressively more weight on abundance of the taxa in these communities, which allow us to both consider rare and abundant taxa along these different parameters. And so kind of jumping into my results, on the bottom we have the effective number of taxa, and I'm going to walk you through it from the top to the bottom. So on the top we have the 2 types of leaf segments that came from leaves that were infected with Rhizoctonia. And so what you can see is that the asymptomatic and symptomatic segments had different fungal communities as it related to taxa richness. So the fungal communities of leaves that were symptomatic of Rhizoctonia had lower taxa richness than the fungal communities of the asymptomatic segments. And when we looked at the other parasites, we didn't see that pattern. You can see that they were not differentiated statistically between the asymptomatic and symptomatic portions of each of the other 2 parasites. And in fact, looking at all of the categories that we included here, the fungal communities of segments symptomatic of Rhizoctonia were the only group that had a difference in taxa richness. And this held across all of the Hills numbers. So as we started to progressively put more weight on abundant taxa, we still saw that there was fewer number of fungal taxa identified in these communities symptomatic of Rhizoctonia. And so to kind of think about these categories of asymptomatic versus symptomatic, we categorize that simply with presence absence. So in the field, we saw a leaf that had a symptom, we say, oh look, there's a symptom, and it would be symptomatic, right? So we would take 2 segments from a leaf like this, the asymptomatic part and the symptomatic part. Whereas a leaf that we couldn't detect any disease by eye, we would call that asymptomatic. However, as you might guess, there are many different types of lesions, not types, well, there are different types of lesions, but different sizes of lesions. And so we wanted to take into account that heterogeneity, so we also estimated disease severity by estimating the percent leaf damaged. And we did this because that's a consideration of disease progression. Smaller lesions are thought to be earlier on in disease progression than ones that are bigger. And so we wanted to consider the relationship between Rhizoctonia severity, this parasite where the fungal communities were less diverse than everything else, and diversity, not just the presence absence. And so if we have, this is just a potential data, if we had estimated disease severity on the x-axis and diversity on the y-axis, we saw a negative relationship between these 2 things. This might be consistent with Rhizoctonia decreasing diversity as it grows through the plant. However, if we saw a positive relationship, it could be consistent with Rhizoctonia disrupting the microbiome upon infection, and then that community recovering in some capacity as the disease progressed. And so looking at my actual data and asking, are we actually seeing if the parasite is progressively decreasing diversity as it grows throughout the plant? Certainly doesn't look that way. So on the x-axis we have disease severity, on the y-axis we have these different Hills numbers. We aren't seeing a negative relationship here. And so this suggests that Rhizoctonia is not progressively decreasing diversity as it grows through the plant. It could be that its initial infection disrupts the microbiome in some way. And so this is just diversity or richness, considering the number of taxa present. But we also wanted to understand not just the number of taxa present, but who's present. So what's the difference in composition, not just diversity. And so here's an NMDS plot where simply, just to kind of go through this, objects that are closer together on the plot are more alike than those that are further apart. And so fungal communities of segments with Rhizoctonia had a distinct composition. So it clustered separately from everything else, where we didn't see that pattern with the other parasites. And so parasite symptoms can have different relationships with host-associated fungal microbiota. And thinking about it, this supported my hypothesis of whether fungal communities of hosts damaged by parasites that modify the host environment might be distinct from the fungal communities of asymptomatic leaves. Well, we saw that Rhizoctonia was associated with a distinct fungal community in those segments, where Puccinia was not, so there was no differences between the fungal community of leaf symptomatic of Puccinia and leaves, I should say leaf segments, and leaf segments of asymptomatic tissue. And so this is in support of my hypothesis. Rhizoctonia is the parasite that's feeding in a way that creates this necrotic tissue, whereas Puccinia is not. Calototricum also had no significant differences in their fungal communities compared to that of asymptomatic segments. And because of this kind of two-phase feeding strategy, it's a little bit more confusing to dissect. And so I think work moving forward that's able to tease apart these different phases and their associations with the fungal community might help us better understand what might be going on here. And I want to add a caveat here that while my hypothesis is trying to establish a causation in these patterns, this is an association study, and experimental manipulations or longitudinal surveys would be needed to establish causation. So parasite traits, here I was considering parasite feeding strategies, might help us understand parasite species-specific results. So there are a number of studies that look at the relationship between parasites and a host microbiome, and they find a range of relationships between parasites and microbial diversity, where some are associated with higher diversity, some are associated with lower diversity. And considering parasite traits may facilitate investigations of why. Why are we seeing these differences between species? So here I considered parasite feeding strategies, but other traits in other systems may be important to consider. So in summary, the microbial community surrounding a parasite can have near-term and long-term impacts. At the individual level, I showed you that within host microbial interactions can affect parasite growth rate, and that higher order interactions or more complex interactions may be important for disease outcomes. At the population level, I showed you a mutualist at the plant level may have a secondary effect on parasite prevalence across host population. And that when we consider the whole fungal community, the relationship between parasites and the host-associated fungal communities may vary by parasite species, or do vary by that's what I showed you. They do vary by parasite species. And so moving forward, I was thinking about kind of what this work together might suggest. It does suggest what a lot of other studies suggest. Things are really complicated. And so the effects of microbial interactions on disease within an individual may not clearly scale up to the population. There may be parasite species-specific interactions with both other microbes and the microbiome more generally. And the direction and magnitudes of interactions may differ under field versus lab-based conditions. So I think moving forward, we need to think about the mechanisms underlying these complexities, which is always the kind of goal of science, I think. And so I want to put forward some things that I think would be really integral in doing this. We need more population-level field-based studies in order to clarify this connection between effects within a host individual and effects across a host population. We need more empirical evidence at the population level. Trait-based approaches may help elucidate these species-specific results. I considered parasite feeding strategy, but considering other traits may be important in other systems. And finally, considering more than just pairwise interactions and considering these more complex interactions, especially including microbes that might be hard to detect in the field, will be really critical for us to understand disease dynamics. Okay, so I'm going to go through my acknowledgements. I went a little longer than I intended. So I'd first like to thank my collaborators who helped in all the projects that I presented on today, as well as the chapter that I wasn't able to include today. I'd especially like to thank Anita and Brandon, who did a lot of the works where I showed their pictures. I'd like to thank my committee. My committee has been so helpful throughout this whole process, both in helping me develop my ideas, giving me things to brainstorm to further help me develop those ideas, thinking about how to most effectively communicate my results to a broad audience, and I really have been very thankful for them throughout this whole process. I'd also like to thank my funding sources, which made all of this research possible. I'd like to thank the biology department as a whole, but specifically I'd like to also thank the third floor Wilson Ecology group, which allowed me to come to their group meetings every other week. It was great to be able to both interact with the results of other labs, as well as think about the best way to communicate my results to a broader audience. I'd also like to thank all of the other folks in the administration and staff here who've been really supportive in various parts of this process. I'd like to thank the Mitchell Lab. So this picture was taken I think my first month of graduate school. This picture was taken last week. I really benefited tremendously from my lab mates. They helped me develop my research questions, especially at the beginning, improve my writing, get me really excited about my research when I was quite literally in the weeds with fieldwork. And I would like to thank Fletcher in particular, who's become a friend and a colleague. We collaborated on a project that wasn't part of this dissertation. I'm really excited to see where that project goes. And of course, I'd like to thank Charles. Charles has been the most amazing mentor I could ask for. He gave me space to develop my own questions, even when they were in directions outside of his own expertise. And I think somewhere on your website at one point it said that you would describe but always around. And I think that you truly stick to that description, where it was really great for me to develop as a scientist by struggling at the beginning, but it was always nice to be able to brainstorm ideas with you and get advice on writing throughout the whole process, analyses I can do, and get excited about the work again. And I think any success that I have moving forward will in large part be due to your mentorship. I'd like to thank the folks who helped in the field. I think there's more grass in this picture than people. These folks have been tremendously helpful, both in this project I mentioned that didn't make it into the dissertation, as well as the pool experiment. This picture right here is actually an emergency harvest of the experiment because one of those tropical storms was coming through in the fall. But seriously, they've been tremendously helpful. I'd like to thank Brooklyn in particular, who spearheaded all the field efforts and has been so tremendous and such a great addition to the lab, both in the field and in the lab. I've been really lucky to meet amazing people in the department while I've been here. I realized as I was making this slide that we pretty exclusively take pictures at our annual tradition of going to a beer and cheese festival. The people that I've met, you know, the time in grad school would have been much less fun without these folks. And knowing that I could count on them to hear me work through ideas or hang out to distract a bit from our work has been really tremendously helpful. We did do things that weren't just beer and cheese festivals, and I did have some friends outside the biology department. They were, you know, a dissertation's a big undertaking, and so I think it really takes a community, both in the department and outside of it. And I think knowing that I could turn to these people just to kind of, you know, vent or to distract myself or work through writing, work through ideas has been really, really tremendous. I'd like to thank Jesse in particular in the middle, who moved down here with me from Boston to North Carolina and really helped me transition to life as a grad student in a new place. And of course, I have to thank my friends who provided support from afar. I moved down here from the Northeast. This is my first time not living in the Northeast, and so most of these friends, I guess at this point, are now in different parts of the country and the world. And so these are pictures of them coming to visit me in North Carolina. No, this is in Savannah. It was part of that trip coming to visit me. And you know, knowing that I could call them on the phone to talk through different things while I've been in grad school has been amazing. I'd like to thank my family who made it down from New York to come see this talk today. I really like to talk on the phone while I walk to places, and they're usually who I call. And so knowing I could talk to them whenever I wanted to, talk about my work, talk about life as a grad student, talk about anything really, has been so, so important And they've been incredibly supportive of my education, but also made sure that I grew up knowing the value of education. I'd also like to congratulate my brother, who beat me to becoming the first Dr. O'Keefe by a few weeks. Finally, I'd like to thank my partner, Mike. We were long distance for most of graduate school, but he was so supportive, even from afar, knowing that he spent a lot of time on planes and on Skype, knowing that I could rely on that support has been really allowed me to get through it all. He even helped in the field once. And this picture is of us finding out that he was matched for residency in Philadelphia. So while we were apart for most of graduate school, we will be together in the next stage. I'm really excited for what that next adventure in Philadelphia brings. And with that, I'll take any questions.
So this is more of a speculative question,
Okay.
but so you did a great job showing the importance of different aspects of the fungal community context.
Yeah.
Do any of these species affect different grasses, or are they all?
Oh, that's a great question. Yeah, that's a great question. So there are different, a lot of cool season grasses have epiploe species like this, have systemic endophytes. Rust in particular, so that that third parasite I mentioned in the final study was specific to tall fescue, but Rhizoctonia is a generalist. So it's actually really devastating to different parts of Southeast agriculture because it really hits root crops a lot. I didn't study it in these other systems, but it would be interesting to see if these patterns help in different hosts. Does that answer your question?
Yeah, and I was also wondering then about like potential, since it may be a fescue dominated grassland, it's not all fescue, or spread between species.
Yeah. Other work in the lab has looked at that to some extent, considering the community, the host or potential host community. So I would suggest looking at that. I have not looked at that though. Yeah.
What's the timescale which Kayleigh switches from this benign to visiting? And would you be able to look at changes in community structure before and after that switch occurs?
So Kayleigh in that first stage is asymptomatic. So in the field, it would be really hard. In the lab, I know I put it there, so a little easier. In the lab, it took about a week and a half to express symptoms, and I should say symptoms here mean that it's shifted into that second phase. But we don't know, while we know that some of what we put there switched to that necrotrophic phase, we wouldn't know if both of them are. But I think someone who's able to tease apart those methods might be able to do that. But in the field, I think it would be really challenging if you wanted to characterize these communities under natural infection. You wouldn't know Kayleigh was present unless someone created a really cool assay that I don't know about until it exhibited symptoms, which shows that it shifted into that second phase. Yeah.
You mentioned that the microbiome study was
Correlative. Yeah.
And before you said that, I was thinking, well, couldn't it be that something about the community that's there enables Rhizoctonia to infect?
Yeah,
and
you're right.
Are there data that
so I think
one way or the other?
Say that again, sorry.
Is there data that might suggest?
I think here, because I collected the asymptomatic part of the same leaf, that might suggest that microbiome wasn't that way. That being said, it still is an association study. I'm not trying to say, yes, I did it, but, but yeah, so I think that's where I was kind of drawing that the parasite is causing something. But work that kind of more longitudinally looks at these populations might be able to tease that apart a little bit better.
Thinking about the diversity of the other fungi, how there are like 40 to 60 other are there. You have like talked about parasitic.
Yeah. So we did go on to try to classify what actually those taxa were. This part of the ITS region, that region that we use to amplify this fungal community, doesn't allow us to get to the species level. And a lot of these genera are filled with a lot of different things, whether they be parasites or mutualists or airborne things or other things. So it's a little hard for us to tease that apart definitively. But I did do that to some extent, where it certainly showed that, for example, between the fungal community symptomatic of rhizoctonia versus asymptomatic part, there was more parasites. So there was more of the parasite in those systems. But I think work that allows to get a finer scale taxonomic resolution would be needed to kind of dig at that a little bit more.
You said epiploe is transferred like to the seeds, right?
Mm-hmm.
So do you think there's another, or can you speculate possibly on the interactions that would maybe occur over the development of the plant? Like I'm not sure when infection occurs for those other two. Do you
So
think that has anything to do with that?
the development of the host
Yes.
as it grows.
Right, and then when they're infected, like as a baby plant versus if they're infected, like.
I know anecdotally, like rhizoctonia smacks young plants, right? So it's pretty devastating parasite. Epiploe is challenging to detect in above ground tissue in younger plants. And so it might be that any effect of epiploe might be more seen in older plants versus younger plants. But all the plants I considered here were all the same age. So I didn't consider that specifically. But I think considering when epiploe becomes detectable in the plant, that's suggestive of when it's actually present in above ground tissue. You might expect to see if it, for example, it inhibited Kayleigh, that you might see that effect more in older plants versus younger ones.
Yeah, so this is really speculative, but how confident are you in these kind of even species level classifications of parasites versus mutualists versus whatever, given the fact that you observed a parasitic, or I'm sorry, a defensive mutualistic species interacting in very strange ways?
Yeah. So I think it can vary on environmental condition for sure, right? So like, for example, if epiploe was a defensive mutualist, but there was nothing to defend against, it is eating off a plant when there's no threat of a parasite or a predator. So it's just being bad for the host in some capacity, right? The host is getting a benefit, but the host isn't getting a benefit. So I think it does vary by environmental condition. In the case of some of these parasites, Kayleigh does change its association with the host. Rhizoctonia can also live in the soil, so it can, it does change to some extent. When it's on a plant, it is most often a parasite though. But I think similar to interactions depending on environmental context within a microbiome, the association with the host would also potentially vary with environmental condition. Yeah. Okay.
First of all, great to
Hello.
see you. So in the baby pool experiment, am I remembering right that like the hosts that were infected with epiploe had a higher biomass and that's what facilitated higher disease spread?
It's not necessarily what facilitated disease spread. That's what I investigated though.
Okay, yeah. So is it possible that the epiploe is acting as a defensive mutualist not by reducing host susceptibility and disease severity, but by increasing host tolerance?
So I did I have this individual level data. So something that I didn't present on today is that as part of that experiment, we also did do these severity surveys on tracked individual leads over time, and it wasn't defensive then either. If anything, it might be the opposite. I don't think it is defensive in relation to rhizoctonia.
All right, let's thank Kayleigh one more time. All right, so we'll now move into her defense committee meeting. In anticipation of a successful defense, I'm planning to host a celebration for Kayleigh at top of the hill after we're done, hopefully at 5 o'clock. I don't intend to send out an email to remind you, so so please join us at 5 o'clock or thereabouts at top of the hill. Thanks again.
I'll be right back.
Yes. Yeah. So we're here. Yeah, so committee. We're in this room. We can do in like 10 minutes.
Can I close this?
Yeah.
Okay. Do you have to log out?
Hold on.
You did so good.
Thanks.
That was great.
Yeah.